Hello and welcome to your video lecture on section 5.4, where we will discuss bulk transport. Now, sometimes our cells are going to need to remove and take in larger molecules and particles, and we're going to need energy in order to do this. So bulk transport is really a type of active transport. Let's get into our learning objectives. We want to be able to describe endocytosis, including phagocytosis, pinocytosis, and receptor-mediated endocytosis, and understand the process of exocytosis. So we'll start off with endocytosis, and again, this is active transport. So we are moving particles that are larger molecules or parts of cells or even whole cells into the cell. And we do this by first having that plasma membrane invaginate and form a pocket, and then we are going to pinch it off and create an intracellular vesicle. So we have a couple of different types of endocytosis. One of them is known as phagocytosis. Now, I've described this process in class before, but it is known as cell eating. So think more solid, larger particles that we're taking in. And again, this is going to involve us to be bringing in the substance using the plasma membrane. Now, we haven't mentioned before that we are going to utilize some proteins that will coat the internal surface of the plasma membrane called clathrin. And once we are able to bring this particle into the cell, then those clathrins are going to disengage from the membrane, and we now would call this a phagosome. And this will eventually merge with a lysosome and start breaking down the particle in there, and the vesicle itself would be called a phagolysosome. Once that breakdown is complete, the contents will be released through exocytosis, and we will describe that exocytosis process at the end of this lecture. Examples of cells that can go through this process of phagocytosis would be our neutrophils or our macrophages, which are both white blood cells that will work to remove invaders such as bacteria, or sometimes they are just gobbling up debris or abnormal particles or really small cells. Next, we have pinocytosis, and the difference here is that it is known as cell drinking. So we're either taking in fluids or smaller particles than compared to phagocytosis. So we'll have a very similar process here where we will see the plasma membrane invaginate and then pinch off into a smaller vesicle. Here we will not have a fusion with the lysosome, but instead we'll just take in that product. We also have this other process that is a type of pinocytosis known as podocytosis, and this is going to utilize something similar to our clathrin, but it is going to be our caveolin, and this is another coating protein that we will see on the membrane's cytoplasmic side, and it helps to bring in small molecules into the cell and possibly transport it to the other side of the cell, which we use the term transcytosis to reference that. This next slide kind of shows you a comparison between these two things. Here is the clathrin-mediated endocytosis, whereas here you're going to see the caveoli surrounding and then eventually fusing with a larger vacuole here to create a cavosome. And you could see they're headed over to the Golgi and the ER. Whereas our clathrin, once that vesicle is formed, it does disappear. There are these little lines through here, so they disappear as it continues to go and attach to a lysosome or has other functions in the body. Now let's move into receptor-mediated endocytosis. We'll see similarities here to what we've already discussed. We will have some receptors along the plasma membrane. You could see it's on the external surface here that are going to have a specific binding affinity for certain substances. The clathrins are down at the inferior side of the I should say interior, not inferior side of the plasma membrane. And if that specific substance attaches to the receptor, it is going to bring that in and pinch it off to create its vesicle, holding the substance still with its receptors. Now, this type of endocytosis is dependent on the receptor. So if the receptors don't match, then the compound's concentration outside of the cell here is going to increase. And that could potentially be damaging for our tissue fluids or our blood that travels to multiple areas of our body. And sometimes we may have different organisms or toxins that are able to attach to these receptors and bring that in. Examples would be the flu virus, diphtheria, or cholera toxin. And then, of course, we would suffer the consequences of bringing that into our cell. Lastly, we're going to talk about exocytosis, and this is when we are moving in the opposite direction. So instead of bringing products into the cell, we are going to expel or move these products outside of the cell into the extracellular fluid. So we would want to do this with waste material that has been put into a vesicle and will move closer to the plasma membrane and eventually fuse with it and then release it out to the external environment. Examples would be secreting proteins that the cell has made or secreting neurotransmitters from one neuron into the synaptic cleft to then be picked up by another neuron. And then this is just a table summarizing some of the transport methods we've talked about, whether they're passive and active, and the material that they transport. So I'll let you look over this, and as usual, if you have any questions, just let me know.